Greetings and welcome to our podcast playlist group assignment. Presenting today we have Annie Boissoneault, Chandler Rondeau, and myself, Julia Chilcott. Our main theme and topic of this presentation is olfaction, everything to do with smell. My name is Julia and this is my portion of the assignment. I fragmented it into small clips and will explain them each. My select podcast was called The Science of Everything podcast by James Bodor, published February 19th, 2014 and accessible by the link seen here. He describes the 35-minute episode as an analysis of the sense of smell and describes the show as a science podcast that conveys accurate and rigorous scientific concepts and findings to a popular audience in an accessible and engaging manner. Here is my first clip.
So, let's get started. Smell, like taste, is a type of chemical receptor. That means that the sensory molecule, the sensory apparatus operates by binding to specific chemicals. This is distinct from, say, the vision, in which sensory transduction occurs by cells that are responsive to photons, light, or other mechanoreceptors, which are receptive to motion. Smell is a type of chemoreception in which the sensory molecules are receptive to particular chemicals. The chemicals themselves are called odorants. If you remember from the previous episode, the molecules that were detected by the taste buds were called tasters. So in this episode we have odorants. Although taste and smell are separate sensory systems in land animals, they are closely related to each other, as we'll see a bit later. And in fact, many water-dwelling organisms often have a single combined chemical sense, which is sort of like taste and smell in combination. Okay, so let's start by talking about the olfactory epithelium. And don't worry, I'm not supposing you already know what that is. I'm going to explain. But smell does not actually occur in the nose. This is probably the single most important thing to understand. The thing that externally we see is the nose, with nostrils and all that, that has nothing really to do with smell. The only thing that does in regards to smell is bring the air into the nasal cavity. Apart from that, the nose does nothing. Smell actually occurs, or the detection of odorants actually occurs in a small sheet of cells at the top of the nasal cavity, which is called the olfactory epithelium. Epithelium just means basically an external or outside layer of cells that's sort of like a thin sheet. So skin, for example, are epithelial cells. In this case, the olfactory epithelium is comprised of specialized olfactory neurons, which do the sensory transduction, which means they actually detect the odorants and pass on the signals to ultimately to the brain. There are also some other cells that comprise the epithelium apart from the olfactory neurons. So one example of these are the basal cells. Basal cells are stem cells, which means they divide to give rise to other cells. So they're capable of division and differentiation. And the basal cells constantly replenish the cells of the olfactory epithelium, replacing them every few weeks. And in fact, the olfactory neuron cells, thanks to the basal cells, are among the only neurons in an adult human which are capable of regenerating. And so there's not exactly understood why this occurs in the olfactory epithelium and not anywhere else, or not in very many other places in the brain. But it's certainly very interesting because it might shed some light on how we can take advantage of this process to assist in treating neurodegenerative diseases like Alzheimer's, for example. If we were able to grow neurons in other parts of the nervous system, that would be a huge breakthrough. But in the olfactory epithelium, it's possible thanks to the basal cells, which are constantly dividing. It also means that if the olfactory epithelium is damaged by inhalation, for example, of toxic fumes or physical injury, or certain types of nasal sprays have actually been known to cause damage to the olfactory epithelium, generally it's capable of regenerating within a few weeks. So usually the damage is only temporary, although in extreme cases it can be permanent. The complete loss of smell is known as an anosmia, and that, as I said, is often temporary as the neurons are capable of regenerating.
This clip discusses how smell is a function of chemical receptors and describes the olfactory epithelium. It relates to the course material because it defines odorants as we learned in Chapter 14, Part 1, which discussed the olfactory anatomy. It details the basal cells, their function, and their unique regenerative ability as seen in the lecture for this part of the course, and as seen in the weekly web activity 14.1 using the Oxford Learning link. Here is the second clip.
Now, each receptor cell has small cytoplasmic projections from its but it sort of divides up into a bunch of small projections that are called cilia. And they are covered by the mucus. So the very outer layer of the olfactory epithelium, the layer at the very edge of the nasal cavity, at the top of the nasal cavity, that is the same layer that is covered with nucleus. The most outer part of that layer is literally the cilia of the dendrites of the olfactory neurons. And on those cilia are the receptor proteins that are able to bind to odorants. Each individual receptor cell, so each neuron, expresses a single, what's thought to be unique odorant receptor. So that is, each neuron has more than one receptor on it, but only one type of receptor, one specific type. And humans are thought to have about 350 of these different types of receptor proteins in total. Rodents have about 1,000. Humans seem to have the remnant or pseudogenes of many of these evolutionarily older olfactory receptor proteins, but they seem to have become non-functional. So if we could find a way of reactivating those genes, we would potentially be able to smell a lot more different substances than we're able to now. But anyway, and these 350 different genes that code for these proteins are scattered all across the genome. It forms one of the largest gene groupings that we know of, all of these different genes for the receptor proteins, because for each different protein that you have expressed, you need a unique gene for that. So each of these 350 different receptor proteins has to have its own gene that corresponds to it. It's important to understand, though, quite different to, say, vision and taste, where there's a fairly distinct mapping between types of, say, wavelengths of light or types of taste molecules and the receptors that detect them. That's not the case in olfaction. In olfaction, there are 350 different types of proteins, each expressed on a different cell, more than one cell, of course. But most of those proteins are able to detect a wide variety of different odorant molecules.
This clip discusses the dendrites, cilia, and exclusivity of the odorant receptors in their corresponding enzymes. It relates to the course material again through the Chapter 14 Part 1 lecture. It lists the number of genes and recalls the sizable portion of the human genome that is dedicated to olfaction. Here is the third clip.
Also, the receptor cell can simply undergo adaptation, which is a general process by which a neuron ceases to respond to a stimulus after prolonged exposure. This is not specific to olfactory cells. It's a widespread phenomenon in pretty much all neurons, that if you see something for long enough, or if you stimulate a muscle for long enough, or if you taste something for long enough, pretty much anything like that, hear something for long enough, the number of action potentials that are fired as a result of that will diminish. Or in other words, the response to a stimulus diminishes over time. It's basically habituation. It's a similar idea. So that happens in the olfactory system as well. It's essentially the same reason. I believe it's called olfactory habituation or something like that. It is the general phenomenon by which, or olfactory adaptation it's also called, by which you go into a room and you notice a smell and it's very distinctive, but within a few minutes you don't notice it anymore because you've become adapted to it. You're essentially, the particular receptor proteins that bind to the odorants that generate that smell have been saturated and have been firing for so long that those receptor cells have become habituated to that stimulus, and so you don't really detect it anymore unless you leave the room and then come back later, and then you can smell it again. Now,
This clip discusses olfactory adaptation. This relates directly to the course material found in Web Activity 14.2 that defines adaptation and its evolutionary benefits.
Well, in the olfactory bulb, the sensory neurons synapse with interneurons or secondary receptor neurons that are found there, but they do so in a particular way. It's not random. There's a particular mapping that's found. In fact, there are sort of spherical clumps of cells in the olfactory bulb that are called, each of which is called a glomerulus, and within each glomerulus, about 25,000 different primary olfactory axons terminate in that glomerulus. And they in turn synapse with about 100 second-order olfactory neurons, which then carry the signals deeper into the brain. So just to try and make that a bit clearer, we have the olfactory bulb, which is a projection of the brain. Within the olfactory bulb, there are a bunch, I think several hundred or maybe a few thousand, I think there are a couple of thousand actually, of these spherical structures called glomeruli. Within each glomerulus, there are a bunch of primary olfactory axons terminate there, and a bunch of different secondary olfactory neurons take their input, so they have their dendrites there, and then project their axons out into the brain. But there is more input than output. So we've got each glomerulus has about 25,000 axons going in, but only about 100 second-order olfactory neurons coming out. So there's a centralization, if you like, of information. There's like lots of small roads running in and only a few big roads running out, if you want to think of it sort of that way. And within each glomerulus, and also within the olfactory bulb as a whole, there's a fairly complex internal network and circuitry structure. So there are interneurons connecting different ways, there's back propagations, and there's inhibition between nearby neurons. There's also some projections that come back from higher regions of the brain, and we don't exactly know what they're for. And we don't fully understand the complex circuit structure here and exactly what processing is occurring, but it is thought that the first sort of lowest levels or first stages of olfactory processing are happening here even as early as the olfactory bulb because of this complex circuit structure. We also do know that each glomeruli receives input primarily from olfactory receptor neurons of a particular type. So if you remember...
This clip discusses the olfactory bulb. It outlines the function of the glomeruli and how they synapse. It relates to the course material through Chapter 14, Part 1 and 2 through verbal explanation of the centralization of each glomerulus and how it aids and starts olfactory processing. This also connects to Web Activity 14.1, describing the connection pattern of sensory neurons to the glomeruli of the olfactory bulb. Here's the fifth clip.
Now, from the olfactory bulb, neurons project back into a large number of higher brain regions. I won't list all of these here. It's actually quite a complicated story, much more complicated than for vision or for taste, where mostly there's just a couple of particular pathways. Here there are many. Basically, the broad pattern, though, is that the neurons from the olfactory bulb synapse with a sort of lower region of the cortex, which is called the olfactory cortex. And from there are projections which run, there's one sort of set of projections which runs into the thalamus and thence into the orbitofrontal cortex. And that is thought to be what the pathway that is responsible for the conscious perception of smells, because the orbitofrontal cortex is the reason that lights up, in fMRI and PET scans and such when we get people to smell things. And that's a region of near the frontal cortex, which is responsible for conscious thought and things like that. So that's all consistent with the idea that this region of the cortex is what's responsible for the conscious perception of smell. But that's by far from the only place in the brain that olfactory information is taken. Remember, I said that pretty much all or many of the neurons from the olfactory bulb project to the olfactory cortex, and from there some of them go to the thalamus. But where do the rest of them go? Well, some of them, some other of the axons project to other regions of the brain. For example, the hippocampus and the amygdala, which are sort of lower regions, part of what's sometimes called the limbic system.
This clip discusses higher brain regions than the olfaction system connects to. This connects to the course material through Chapter 14 in the Olfaction Part 2 lecture, defining the path from the olfactory receptors to different parts of the brain. Lastly, here's the sixth small clip.
Quite interesting. So, as I've said before, many animals have a much keener sense of smell than humans. So, as I've said before, many animals have a much keener sense of smell than humans do. Broadly speaking, humans have very good sense of vision, particularly colour vision, compared to most animals, but a fairly poor sense of smell compared to most animals. Most mammals in particular have quite good sense of smell, humans being a sort of relative exception, whereas most birds do not have a very good sense of smell. A lot of fish also have a fairly good sense of smell, and many fish also, or water-dwelling animals in general, have taste and smell chemoreceptor sense, as I mentioned earlier. Dogs in particular have quite a good sense of smell. It's estimated that, in general, dogs have an olfactory sense about 100,000 times as acute as a human's. You may have heard figures like this before. It's very important to understand what they mean. The way they test this is basically by repeatedly reducing the concentration of an odorant molecule and seeing if the animal or the human can detect it. So you present them with a high concentration and then reduce it a bit, maybe by a factor of 2 or a factor of 10, see if they can still detect it. If they can, you reduce it more and you reduce it more, until you reach the stage where it's too dilute for them to detect the odorant molecule. There's not enough of it left to register. And that's how sensitive the organism is said to be to that particular type of smell. And so in doing experiments like this, dogs, on average, come out as being able to detect concentrations 100,000 times lower than humans are able to detect. That doesn't mean that they smell everything 100,000 times as intensely as a human does. So it's not like chocolate smells 100,000 times more like chocolate or more chocolatey than to a dog than it does to a human. Or feces smell 100,000 times worse to a dog than a human. That's not a good way of thinking about it. Probably these things smell, well, they wouldn't smell exactly the same. But I suspect the intensity of something like that is not dramatically different. It's just that they can detect much, much lower concentrations than a human can. So that's dogs in general, but there are a type of dogs that are specifically bred for having a particularly acute sense of smell. The general term for this is scent hounds, and the most famous example are bloodhounds, which were developed in Britain in the early modern period. There's a little bit of controversy exactly how, or what their origin was, but anyway, they've been around for a few hundred years, and they have the keenest sense of smell of any dogs, as far as I know. They have noses that are some 10 to 100 million times more sensitive than humans, so that's about 100 times more sensitive even than most dogs. Again, this doesn't mean that every smell is ridiculously overpowering to them. It means they can detect much lower concentrations of things than humans can. They've been known to be able to detect trails of people's from only a few cells. So that means a very, very small number of molecules are actually entering their nose there. So that's very impressive. And that's essentially why they were bred. They were bred basically to be hunting dogs, and now they're used in police investigations and a variety of other things. And they're specially trained to be able to detect and follow trail sense. There are some amazing reports of the feats that they're capable of doing, like of following sense that are trails that are several days old, even across water and vast distances and other things like that. So it's their very dense concentration of olfactory receptor neurons that is that plus their very strict training, which allows them to do this. A bloodhound's nasal chambers are much larger than those of most other breeds, and they have about 4 billion receptor cells, which, if you remember, is about 1,000 times more than a human's, who have around 5 or 6 million. So much, much larger number of receptor molecules, which allows them to be so good at following these types of scent trails.
This clip discussed other animals' heightened sense of smell. This relates to the course material directly through Chapter 14 in the first olfactory lecture, where it is mentioned that dogs have 100 times more OSNs and more brain area that is dedicated to olfaction. Indirectly, it adds an interesting element of understanding to the course material through a clear explanation and appreciation of multiple different forms of mammal olfactory systems. Now on to Chandler's portion.
The podcast that I listened to was the podcast series called The Smell Podcast. I'm specifically looking at episode 2 of The Smell Podcast. This podcast follows someone who lives with anosmia, which is the loss of the ability to detect one or more scents. This alone shows its connection to the material covered in class, being that we did learn about the olfactory disorders. The segment I'm going to be focusing on is the first segment listed, which is from 22 seconds to 6 minutes and 5 seconds. In this segment of the podcast, she explains a specific case. Something interesting about the case is that it explains how smell and taste interact to create flavor. They mention that our taste receptor cells interact with the retronasal olfaction as one way to produce flavor. Since individuals with anosmia have lost some or all ability to detect scents, this causes there are several nerves that transmit sensations of taste, it is difficult to have something take them all out. Olfaction, however, uses only one nerve and thus is much easier to knock out. From head injury or viral infection, although due to the constant replenishing of olfactory receptors, it is possible for olfaction to be restored after damage from a viral infection. Then the second portion of this podcast that I'll be focusing on specifically is from 7 minutes and 20 seconds to 10 minutes and 35 seconds. In this portion of the podcast, she ended up mentioning another olfactory disorder called parosmia. This is pertinent because we did cover olfactory disorders, as I mentioned above, and / what occurs when the olfactory system malfunctions. An individual with parosmia will typically be able to detect scent fairly well. However, they will detect scents that are not present. These scents are often referred to as phantom smells. An odd yet interesting component to parosmia is that as we age, typically our ability to detect scents lessens in people with parosmia. However, their ability to detect scents decreases, but their detection of phantom smells increases as they age. A study found that parosmia is more common amongst women than men, and actually more common amongst younger women than younger men. It was also found that within the same study that parosmia is more likely to occur in those of lower socioeconomic status. This is largely due to them being more likely to be exposed to air pollutants or different illnesses because of lack of being able to afford different healthcare or having less ability to afford a healthier lifestyle. And then another interesting thing was that anosmia, is similar to parosmia, because they can both result from a head injury. Overall, that was kind of the gist of the podcast. Aside from within the specific case, she does explain more of a story that goes along with it. However, that story is not pertinent to the actual content that we have covered. And so that would be all for my portion.
Here's our first story. The name of this article is Medical Device Maker's Malady Inspires Restaurant to Offer Tasting Dinner for Those Who Can't Smell. It was written by Barrett J. Brunsman, who is a staff reporter for the Cincinnati Business Courier. The article was published on August 17, 2018. James Shorey, the Chief Technology Officer for Control Bionics in Milford, has spent 30 years designing and marketing medical devices that help people cope with disabilities. But the inventor discovered there is no cure for his anosmia, which is a disorder that deprived him of the sense of smell. Few people realize the importance of the sense of smell until it's gone, Shorey said. Anosmics are susceptible to depression. They seem to have a higher incidence of social anxiety. They might fret because they can't smell their own body odor. They can't detect by smell whether food has spoiled. They may face dangers such as not being able to notice the smell of smoke, which warns of a fire. Imagine gathering with friends at a restaurant and not being able to share in the enthusiasm for the aromas wafting from the plates of creative cuisine. Add in that without the sense of smell, everything tastes bland. The sense of taste is a mere shadow of itself without smell, Shorey said. And for many, this inability to really taste food is the most significant loss. However, Shori's experience with anosmia proved inspiring to Paul Baracco, chef at 20 Bricks and Milford. The restaurant planned to hold a 5 tastes dinner on Thursday night, which was to feature course selections designed to appeal to anosmics. The courses were to explore the 5 basic tastes detectable by people, regardless of whether they have a sense of smell: sweet, salty, sour, bitter, and umami. Each course was to be paired with an adult beverage to enhance the palate. The special tasting dinner sold out in advance, Shori said. A few anosmics were to gather with family and friends for the occasion. The chef's dishes would promote a feeling of thanks and fellowship, Shori said. It was to be a gastronomic discovery of sorts, Shori said. The food will appeal directly to that real estate on our tongue and soft palate, upper esophagus, cheek, and epiglottis, and it is sure to be delicious to everyone, including to the 1 or more anosmics seated at that 20 Bricks table. Anosmia is, without doubt, the least researched and least understood of the conditions that affect our senses, Shori said. Everyone understands blindness and deafness. Close your eyes, plug your ears, but try to explain to someone what it is to be an anosmic, and there is often a complete lack of understanding and compassion. 2 years ago, Shori said, he suspected that his long-time diminished sense of smell had completely left him. That was confirmed during an appointment with Dr. Alan Seaton, a professor of otolaryngology, head and neck surgery at the University of Cincinnati College of Medicine. Seaton, a UC Health doctor who is director of the Taste and Smell Clinic, was invited to be the featured speaker at the dinner. Side note, otolaryngology is not just a hard word to say, but it means the branch of medicine that deals with the anatomy, function, and diseases of the ear, nose, and throat. Okay, back to the story. One of only a few doctors in the nation with expertise in taste and smell disorders, Seaton agreed to speak to help people understand how important smell is and the difference between smell and taste. The flavor of the food is based on what we're tasting, but our taste buds really only respond to salt, sour, sweet, bitter, and umami, which is a protein-type amino acid kind of meaty taste, Seaton said. Of course, there is the texture, temperature, and look of the food, but the main ingredient of flavor is what we're smelling as we eat. We get odors that are released by the food that come up behind our nose through our throat. What I have people do is hold their nose and put a jelly bean in their mouth and start chewing, Seaton said. They can tell it's sweet, but not much else. Then you let go of your nose, and suddenly you can tell it's cherry flavor or whatever. You couldn't get that without being able to smell it. Often, when people lose smell, they don't even recognize that they lost smell, Seaton said. They say, I can't taste anything. Everything is bland. Everything is flat. They are actually tasting fine. They can tell something is salty or sweet. They just aren't getting the flavor because they aren't smelling. It's rare to lose your taste, Seaton added. We have 6 different nerves for taste buds, so it's rare to have something knock out all 6 nerves. With smell, we only have 1 nerve. Up to 20 million people in the nation might suffer from anosmia, Seaton told me. The most common reason is a viral infection such as a cold or ongoing chronic sinus inflammation, which can result in partial or complete loss of the sense of smell. The disorder can also be caused by a head injury that stretches or tears the olfactory nerve. Medicine isn't effective on viral infections, but might help with sinus inflammation. However, if the olfactory nerve that sends messages to the brain is torn as the result of the brain sloshing around inside the head following a blow to the body or whiplash, there is no medicinal remedy. The good news is the nerve endings undergo constant degeneration and regeneration during our lifetime, so there's the potential that after an insult such as a viral infection or even a head injury that they could possibly regenerate, Seaton said. If regeneration doesn't occur and the sense of smell doesn't recover, the biggest issue is quality of life. It just takes all the fun out of eating, Seaton said. We do it socially. Shori has a professional interest in anosmia as well as a personal one. His work as a designer of medical while having a smell and taste disorder. Our next story is about parosmia. I don't don't suffer from this condition myself, but it impacts a lot of people. Parosmia is a disorder of the sense of smell, especially the perception of odors that are not present, aka it's having phantom smells. The title of this article is "This is the Part of Aging That Really Stinks," and it's written by Robert Preet. The article was published on Thursday, August 16, 2018 in Health Day News. Unpleasant phantom odors haunt many older Americans, a new study finds. Of more than 7,400 people over age 40 who took part in a federal health survey, 6.5% said they experienced nasty odors, such as burning hair or the reek of an ashtray, from nowhere. That's 1 in 15 people. As folks age, their ability to identify odors tends to decrease, but their detection of phantom odors increases. Why this happens is a mystery, but smelling something that isn't really there can be life-changing, the researchers said. Problems with the sense of smell are often overlooked, despite their importance. They can have a big impact on appetite, food preferences, and the ability to smell danger signals such as fire, gas leaks, and spoiled food, said Judith Cooper. She's acting director of the U.S. National Institute on Deafness and Other Communication Disorders, or the NIDCD, which led the study. Lead researcher Kathleen Bainbridge said overactive odor-sensing cells in the nasal cavity or a malfunction in the brain area that understands odor signals may be involved. The new study lays the groundwork for further research. A good first step in understanding any medical condition is a clear description of the phenomenon. From there, other researchers may form ideas about where to look further for possible causes and ultimately for ways to prevent or treat the condition, Bainbridge said in an Institute news release. She is an NIDCD epidemiologist. Study co-author Dr. Donald Leopold is a clinical professor of surgery at the University of Vermont Medical Center in Burlington. He said many people who experience strong phantom odors have a poor quality of life. They may also have trouble maintaining a healthy weight. The new research follows a Swedish study that reported 4.9% of people older than 60 experienced phantom odors. It said the rate was higher among women than men. This new study found a similar rate among Americans over 60, but an even higher rate among those between 40 and 60. Roughly twice as many women as men reported experiencing phantom odors, and the gender gap was greatest in the younger group, the NIDCD study found. Besides gender, other risk factors for experiencing phantom odors include head injury, dry mouth, poor overall health, and being poor, the researchers said. They said poor people may have greater exposure to environmental pollutants and toxins or have health conditions that contribute to the problem, either directly or because of medications they take. This study was published on August 16th in the journal JAMA, Otolaryngology Head and Neck Surgery. Do any of you listeners suffer with parosmia or know someone else who does?
Now on to Annie's portion.
For this assignment, I used the podcast Totally Cerebral, What's That Smell? On this episode, Dr. Wendy Suzuki spoke with neuroscientist Howard Eichenbaum about his experience in studying how the memory works using olfactory stimuli. Howard began his studies with rodents, since they are known to remember smells extremely well. The study consisted of mixing certain household odors, such as oregano and cinnamon, mixed with clean sand and burying a food reward at the bottom of one. For this, the food reward was at the bottom of the mix of sand and oregano odor, and when the rodents would sniff it, it would take them only one try to learn which of the mix was good. Along with this, they were able to remember for at least 1 week which was the correct mix, even when adding new mixes of sand and odor. To further their understanding of rodents' memory capacity, they examined their communication by trying to understand their ethology, their normal lives, and how they investigate the world. They found that rodents are way better at using olfactory stimulation compared to humans when it comes to finding rewards. It is well known that rodents are foragers. In this sense, part of the colony will go out and retrieve food and come back to the group where the remainder of the rodents will sniff them on the return. The food they have eaten will mix itself with the smell of their breath, called carbon disulfide, and when presented to another rodent, they will automatically prefer that odor and follow the forager for food. This is also a way to test to see whether the food will kill the rats or not. If the rat comes back unharmed, it is a good sign and the colony will follow the ability of rodents to use their olfactory system is extremely important in their survival. Humans, on the other hand, are not as great at smelling things or following smells since we do not have a snout, the specific part of the olfactory system that works wonders at picking up odors from the ground. However, what we do have is retronasal apparatus. This means the smell coming from something you are eating will travel to the back end of your nose, which is in some way as good as animals. This is also where the majority of flavors come from, since the smell accentuates it. Now let's focus on the human olfactory system and how it can affect our memory and bring us back to a specific time in our lives. The power of an olfactory stimuli is incredible for remembering past events, as Chef Anita Lowe will demonstrate with her earliest memory going back to the age of 2. How is this possible? Wendy sat down with Kent Kirschenbaum, a chemist, to learn how food plays with our senses. He explains that the olfactory system can easily and directly access our emotional and memory processes. The olfactory system has an immediate access within one connection, or synapse to be precise, to the temporal lobe system, more particularly the entorhinal cortex. The entorhinal cortex is a hub of network for the memory, navigation, and the perception of time. Part of this cortex will project to the hippocampus, which is a crucial part of our brain for memory. More specifically, it is projected to a part of the hippocampus, which is responsible for resurrecting context. As we can see, compared to many other sensory systems, the olfactory system has less of a process before reaching the memory centers or the emotional centers of the brain. Not only does the course of the olfactory pathways have a direct impact on the temporal lobe, but they also directly impact other structures that have an emotional impact. Such other structures being reached are the amygdala, responsible for our emotion, as well as the part of the hypothalamus, having a lot to do with affect. These are the first pathways to be developed and will come into play immediately, while at the other end of the spectrum is the memory pathways, since we build our memory around things we can remember about odors and what they might be associated with. The last to develop is the pathway access from the olfactory system to the frontal lobes. Particularly, the pathway leaves parts of the frontal lobe that judge the value and quality of odors and help us decide on what smells good and on what combinations work together. The olfactory system connections is also a playground for chefs, such as Anita Lowe, who uses her memories and experience to create her most famous dishes. After years of practicing and learning the art of cooking, your olfactory system will be refined with all of the enrichments you have learned through the years. Everyone has the capacity to expand their smell, since it is similar to muscles in the brain. The more we try and the more we experience, the more we create new cells in the olfactory bulb and refine our capacity. The more that we try and experience, the more that we create new cells in the olfactory bulb and refine our capabilities. Since smell has a direct pathway to the emotional centers of our brain, we are able to relive many memories from our past.
The audio for this podcast was not downloadable, but can be found at the link provided, and the clips can be found with the provided timestamps. That is all. Thank you for watching and listening.